So our topic is beta-lactam antibiotics. We will talk about the structure, mechanism of action and the classification of beta-lactams. So what are beta-lactam antibiotics? Right? So they are the most commonly used antibiotic class worldwide. Right? If you take anywhere in the world, 60% of the world's antibiotic usage is beta-lactam antibiotics. Why do you call them beta-lactams? Because they have a beta-lactam ring, a 4-membered ring in their structure. We call them beta-lactams. Right? Then there's a side chain attached to the beta-lactam ring that determine the different properties of the beta-lactam. We call penicillins, cephalosporins, carbapenems, right? Monobactams. All the beta-lactams, the structure is determined by the structure varies, especially in the structure of the side chain. So this is the diagram of a beta-lactam ring. You can see 4-member ring, right? So you can see this area, right? So this is the bond that beta-lactamase attack. Beta-lactamases are enzymes produced by the bacteria that destroy beta-lactam antibiotics. They attach to this bond. This is the bond between carbon and nitrogen bond. The bond between carbon and nitrogen, right? So that's why that is where the bacteria beta-lactamases work. And this is the side chain. So this is the side chain, right? So this side chain, the structure of the side chain determines the the properties of different beta-lactams, right? Penicillins, cephalosporins, carbapenems, monobactams, right? They all differ. The major difference is in the structure of the side chain. The beta-lactam ring, there is another ring here. We'll talk about those things with different classes of antibiotics. So this is the structure of beta-lactam ring. Right. So how beta-lactam, where do they act? They act on the peptidoglycan. So you remember the peptidoglycan, that is the major component of the structure of the cell wall. Staphylococcus aureus, the gram-positive organisms such as Staph aureus, the peptidoglycan layers, there are about 10 layers of peptidoglycan in their cell wall. But gram-negative such as E. coli, there are about 2 layers. Right, so there are more peptidoglycan in gram-positives than the gram-negatives. That is why the beta-lactams act on the peptidoglycan. So beta-lactams are very effective against gram-positives more than the gram-negatives. Right. So first we'll talk about the structure of peptidoglycan. So you can see alternative molecules of N-acetylglucosamine, N-acetylmuramic acid, N-acetylglucosamine, N-acetylmuramic acid. They are linked by beta-1,4 linkage. This is the backbone of the peptidoglycan, right? It's a the carbohydrate backbone, right? Then E. coli, gram-negatives also have the same backbone, same structure, right? Then there is a pentapeptide chain attached to the N-acetylmuramic acid molecule. Right. In the case of gram-positive bacteria, this is L-alanine, D-glutamic acid, L-lysine, D-alanine, D-alanine. That is the pentapeptide side chain attached to N-acetylmuramic acid. All the N-acetylmuramic acid, they have the pentapeptide side chain. Right. So this is another chain. Right. So in the case of gram-negative bacteria, it is the same peptide except the third amino acid is not L-lysine, it is mesodiamino-pymalic acid. Mesodiamino-pymalic acid, that is the amino acid in the third position in gram-negatives. Other than that, they are the same. This is gram-positive bacteria, it is L-lysine, gram-negative is it mesodiamino-pymalic acid. In addition, in the case of Staph aureus, there is a pentaglycine attached to third amino acid, that is glycine 5 molecules attached to L-lysine. Right? So those are the small differences between gram-positive and gram-negative pentapeptide chain. So now what happened? Now the peptidoglycan chains, these peptidoglycan chains are not strong enough to make the cell wall, to give the cell wall the strength that they need. Right? The strength is provided by cross-linking between the peptidoglycan chains. The cross-linking occurs between the pentapeptide chains. You can see this chain cross-link with this chain. So there are multiple chains, right? So they cross-link with each other. This makes the peptidoglycan layer strong. If there is no cross-linking, if the cross-linking is inhibited, right? So the peptidoglycan layer is not strong, and so the water will enter. And the osmosis occurs, right? Water will enter when the peptidoglycan layer is not strong, water will enter the cytoplasm and the cytoplasm the cell undergo lysis, right? So that is what happened when the peptidoglycan becomes weak, right? So now this cross-linking is, what is the enzyme that induces cross-linking, that catalyzes cross-linking? Transpeptidase. Transpeptidase is the enzyme that catalyzes the cross-linking, right? Transpeptidase. So beta-lactams bind to this transpeptidase and inhibit the activity. That is where where does the beta-lactam cause effect? They acts by bind to the transpeptidase and by inhibiting its activity, right? So that is why we call them penicillin binding protein. Why penicillin binding protein? It is all the beta-lactams, not only penicillins, right? They all bind to the transpeptidase, but penicillin was the first antibiotic to be discovered, right? So those days we started to call them penicillin binding proteins. Later on other antibiotics were discovered now, so but we still call them penicillin binding proteins, right? Remember that is the site of binding for all the beta-lactams, transpeptidase. So when the transpeptidase is inhibited, no cross-linking, peptidoglycan layer is not strong, so the water will enter, cells undergo lysis, that is lysis by osmosis, right? So that is how the beta-lactam antibiotics work, right? Cells undergo lysis. So that's why beta-lactam antibiotics are bactericidal. Bactericidal. They kill the bacteria. Bactericidal antibiotics, right? So this is the in details. So peptidoglycan, which is an important component of both gram-positive and gram-negative cell walls. Backbone of each peptidoglycan chain is made of alternating units of N-acetylmuramic acid, N-acetylglutamic acid, joined by the beta-1,4 linkages. Peptidoglycan layer of the cell wall is made strong by extensive cross-linking between the peptidoglycan chains. Cross-linking between the chains occur via pentapeptide chains attached to the N-acetylmuramic acid. The cross-linking is catalyzed by the enzyme transpeptidase. Beta-lactams bind to the transpeptidase and inactivate by acylation. Transpeptidase is popularly known as penicillin binding protein since the penicillin was the earliest beta-lactam to be discovered. Without cross-linking between peptidoglycan chains, the cell wall is weak and allows osmotic entry of water into the bacterial cells, resulting in cell lysis and bacterial death. Therefore, beta-lactam antibiotics are bactericidal antibiotics, right? So these are all the beta-lactams that we need to talk later on in separate lectures, right? Penicillins, beta-lactamase inhibitor combinations, right? Like amoxicillin clavulonate, right? Ticacillin clavulonate, piperacillin tazobactam. So we'll talk about those things. Cephalosporins, monobactams, carbapenems. We'll talk all these antibiotics in separate lectures. Remember, it is very important to have a very good knowledge about antibiotics, right? So because that is going to help once you if you are a nurse or a pharmacist or a doctor. Once you enter your profession, the knowledge is going to be very helpful. Thank you. I hope you enjoyed the lecture. Thank you. If you found this video helpful, please like this video and subscribe to my channel. You can get new video notifications by clicking the bell icon. My Patreon supporters can request lectures and get exclusive content. Please check the description for links and more details. Thank you.